In 1902, Rudyard Kipling published a story called How the Leopard Got His Spots. The leopard, in Kipling's telling, pressed its fingers against its skin while hiding in a forest, leaving dark marks that became permanent. It was a myth, dressed up in the playful, explanatory style Kipling used for his Just So stories. Stories that asked how things came to be and answered with charming, invented logic. Everyone reading it knew it was not science. But for decades after Kipling wrote it, the actual science was not much better. Naturalists could describe leopard spots in precise detail. They could measure the rosettes, count them, compare them across subspecies, note that Amur leopards had larger ones and Arabian leopards had denser ones. They could observe that leopards living in forest had denser, smaller rosettes, and leopards living in open grassland had larger, more spaced ones. They could point to the fact that no two leopards have identical rosette patterns, the same way no two humans have identical fingerprints. What they could not do was explain the mechanism. Why do spots form at all? Why do they space themselves evenly across the body? Why do some cats have spots and others have stripes? And what determines which pattern a given species gets? Why does the same individual animal sometimes have spots on its flanks and vague streaks near the spine? These questions sat unanswered for the first half of the 20th century, because answering them required a type of biology that did not yet exist, and a type of mathematics that had not yet been applied to animals. The first person to propose a real answer was not a biologist. Alan Turing is remembered for breaking the Enigma code during the Second World War, for his foundational work in theoretical computer science, and for the test that bears his name for evaluating machine intelligence. In 1952, 2 years before his death, he published a paper in the Philosophical Transactions of the Royal Society titled The Chemical Basis of Morphogenesis. The paper was about how biological structures, organs, limbs, and yes, coat patterns, could emerge from the interaction of chemicals in a developing embryo without any external blueprint directing them. Turing described a system with 2 components: a chemical that activates a process, say pigment production in skin cells, and a chemical that inhibits the same process. The activator stimulates its own production and also stimulates the inhibitor. The inhibitor suppresses the activator. If the inhibitor diffuses faster through the tissue than the activator, if it spreads farther, faster, then a self-reinforcing spatial pattern emerges from the interaction. Wherever the activator briefly dominates, a spot or stripe of pigmentation forms. Wherever the inhibitor sweeps in and suppresses it, the surrounding skin stays light. The pattern is not drawn, it calculates itself. Turing showed mathematically that this reaction-diffusion system could produce spots, stripes, rings, and spirals depending on the relative diffusion rates of the 2 chemicals, the size of the tissue on which they operate, and the timing of when the system activates during development. A faster inhibitor relative to the activator produces spots. A slower one produces stripes. The same basic mechanism with different parameters generates both. Applied to a developing embryo, specifically to the skin of a felid fetus during the period when pigmentation patterns are being established, the model predicts exactly the kinds of patterns we see in cats. It predicts that larger animals should have larger spots, because the same diffusion process operating over a larger tissue area produces patterns at a larger scale. It predicts that the tail, being a narrow cylinder, should produce stripes rather than spots even in spotted animals, because the geometry of a narrow surface favors striped solutions to the reaction-diffusion equations. Both of these predictions are true of real leopards. The spots are larger on bigger subspecies. The tail tip often shows banding rather than clean rosettes. Turing's math predicted this 50 years before the genes encoding the activator and inhibitor were identified. Perhaps the most striking prediction was about animals that are striped on their body but spotted on their legs, or vice versa. Turing's model said this could happen if the pattern was established at different developmental stages on different body regions, with different timing producing different solutions to the same equations. Cheetahs have solid spots on their body and a mane of longer fur along the back of the neck that runs in streaks rather than spots. Exactly the kind of regional variation the model predicts when different parts of the body surface undergo patterning at slightly different times. The molecular biology caught up in stages. Studies of domestic cat coat patterning, which showed the same range of patterns from mackerel tabby stripes to spotted tabbies to blotched tabbies, provided a tractable experimental system for identifying the underlying genes. A landmark 2012 study by Christopher Kalin and colleagues, published in Science, identified a gene called TACPEP, transmembrane aminopeptidase Q, as a key controller of tabby patterning in domestic cats. Cats with one version of TACPEP had mackerel stripes. Cats with a different version had blotched or marbled patterns. The gene was acting precisely as the activator in Turing's framework, setting the spatial frequency of the pattern. Follow-up work identified the inhibitor, a secreted protein called DKK4, encoded by the DKK4 gene, which diffuses more rapidly through developing skin than the activating signal and suppresses pigmentation outside the activated zones. The activator and inhibitor in the Turing system were not abstract chemicals. They were specific proteins encoded by specific genes with specific expression profiles during fetal skin development. Turing's equations described the system accurately. The genome stored the instructions for building it. With the molecular machinery identified, researchers could examine variation in coat pattern genes across wild felid species. The results confirmed what Turing's model predicted and added specificity that the pure mathematical approach could not provide. Leopard rosettes differ from cheetah spots not because leopards and cheetahs are running different patterning programs, but because the same Turing mechanism is producing different output based on differences in the timing and spatial scale of the developmental window. Cheetah spots are solid, filled circles of dark pigment. Leopard rosettes are hollow, rings of dark pigment enclosing a patch of lighter, tawny background. The difference corresponds to a difference in how long the activator signal dominates the center of each emerging spot before the inhibitor catches up and suppresses it. In cheetahs, the inhibitor catches the center quickly, filling the spot solid. In leopards, the inhibitor arrives later or more weakly at the center, leaving a tawny interior ringed by dark border. One gene difference in the timing of the inhibitor's diffusion changes the visual output of the entire coat from solid spots to rosettes. This is not a trivial cosmetic distinction. The rosette pattern creates a more complex visual disruption of the animal's outline than a solid spot does. The hollow center adds a second layer of contrast that makes the shape harder to resolve at a distance against dappled vegetation. A rosette is a more sophisticated camouflage unit than a solid circle, and the genomic data suggests it has been under strong selection in leopard populations living in forested environments where the ability to remain invisible while approaching prey determines whether an animal eats. Melanism in leopards, the all-black coloration of the animals commonly called black panthers, is produced by a different but related pathway. Melanistic leopards carry mutations in the gene encoding the agouti signaling protein, abbreviated ASIP. In normally patterned leopards, ASIP is expressed in the light zones between rosettes, where it binds to the melanocortin-1 receptor on pigment-producing cells and switches them from dark eumelanin production to lighter pheomelanin. The rosette borders stay dark because ASIP signaling is suppressed there, allowing eumelanin to dominate. In melanistic leopards, mutations in ASIP or in its receptor eliminate this switching mechanism across the entire coat. Every pigment cell defaults to eumelanin production, regardless of its position relative to the Turing pattern. The Turing pattern itself is still there. Melanistic leopards still carry the patterning genes, and in certain lighting conditions, the ghost of the rosette pattern is visible in the coat as slightly different intensities of black. Photographers working in the forests of Southeast Asia, where melanistic leopards are most common, have captured images in infrared light that make the ghost rosettes clearly visible. The same pattern as a normal spotted leopard, rendered entirely in 2 shades of black. The spots did not disappear. The color switch that makes them visible was turned off. A 2010 study by Eduardo Iziric and colleagues confirmed that melanism in leopards is caused by loss-of-function mutations in ASIP, and showed that the same gene is responsible for melanism in at least 6 other felid species: jaguars, servals, pumas, jaguarundis, Jeffrey's cats, and oncillas, with the mutations arising independently in each lineage rather than being inherited from a common melanistic ancestor. Melanism has evolved over a dozen separate times in felids alone, always through mutations in the same handful of genes in the same pigmentation pathway. Evolution keeps finding the same molecular solution to the problem of producing a black coat, because the pathway is constrained. There are only a few places in the signaling cascade where a loss-of-function mutation produces all black coloration rather than lethality or a patchy coat. Jaguars present an especially interesting case for comparison with leopards, because the 2 species have rosette patterns that look superficially similar. Both are hollow rings of dark pigment on a tawny background, but differ in one consistent way. Jaguar rosettes typically contain 1 or more dark spots inside the ring, while leopard rosettes are empty. This difference corresponds to a small but measurable difference in the Turing parameters of the 2 species, producing a more complex patterning output in jaguars. More important than the difference is the similarity. Jaguars and leopards are not each other's closest relatives. Jaguars are the only living member of their lineage in the Americas, while leopards are an Old World species. Their last common ancestor lived approximately 6 million years ago. The rosette pattern evolved independently in both lineages, not because they inherited it from a common ancestor who had rosettes, but because the same Turing mechanism, operating on similar body plans and similar ecological pressures for camouflage in dappled light forest environments, converged on a similar solution. 2 separate genomes on 2 separate continents ran the same mathematics and arrived at nearly the same visual output. The pattern is not a family resemblance. It is a convergence proof. The Amur leopard, Panthera pardus orientalis, is the northernmost and most genetically isolated subspecies of leopard, living in the temperate forests of the Russian Far East and northeastern China. Its rosettes are noticeably larger and more widely spaced than those of African leopards, an observation that has been documented in natural history literature for over a century. The genomic explanation for this pattern difference follows directly from Turing's model. Larger body size and a different thermal environment during fetal development shift the spatial scale at which the reaction diffusion system operates, producing larger pattern elements at lower density. The Amur leopard's coat is also paler and thicker than African leopard coats, adaptations to a climate that can reach -30 degrees Celsius in winter, and these differences in background coloration interact with the patterning system to produce an animal that looks markedly different from its equatorial relatives despite sharing the same fundamental patterning genes. The genome encodes the program. The environment during development sets the parameters. The Amur leopard genome is also the record of a population in catastrophic decline. The species fell below 100 individuals in the wild by the early 2000s, making it the rarest wildcat on Earth. Rarer than the Amur tiger, rarer than the snow leopard, rarer than the cheetah. Population genomic studies of living Amur leopards show the consequences of this bottleneck in the genome itself. Reduced heterozygosity across the genome, runs of homozygosity, long stretches of identical DNA inherited from both parents that indicate recent inbreeding, and elevated frequencies of recessive deleterious alleles that would normally be purged from larger populations. The genetic diversity of the Amur leopard population is significantly lower than that of African leopard populations, lower than captive leopard populations managed with stud books, and approaching the levels seen in the Florida panther before the genetic rescue intervention of the 1990s brought in 8 Texas pumas to restore diversity. A population this small and this genetically uniform is vulnerable not just to demographic accidents, a disease outbreak, a string of bad winters, a poaching surge, but to the slow genomic erosion that occurs when harmful recessive mutations cannot be weeded out because there are not enough unrelated individuals to produce the heterozygous offspring in whom those mutations would remain hidden. Recovery programs for the Amur leopard have involved both habitat protection in Russia and China and managed breeding in captivity, guided in part by genomic data identifying which individuals are least related and which pairs would maximize the restoration of genetic diversity in offspring. Camera trap surveys in recent years have counted over 100 individuals in the wild in the Land of the Leopard National Park in Russia, a number that suggests stabilization, if not yet genuine recovery. But the genomic signature of the bottleneck will remain in the population for generations, regardless of how many individuals survive. What makes the leopard story unusual in the context of animal genomics is that it operates on 2 completely different timescales simultaneously. The genomic basis of coat patterning is ancient. The Turing mechanism and the core genes encoding it predate the divergence of the living felid lineages and run on essentially the same molecular machinery in lions, tigers, cheetahs, domestic cats, and leopards. The variation in rosette size, pattern density, and melanistic frequency across leopard subspecies is recent. It tracks the last few 100,000 years of population history, climate shifts, and local ecological pressures. The ancient program generates the structure. Recent evolutionary forces tune the parameters. A melanistic mutation that arose once in an African leopard population and was maintained by selection, possibly because black coat coloration improves camouflage in dense forest under low light conditions, spread through that population's descendants but not others, producing the regional variation in melanism frequency we see today. In Southeast Asian forests, where dense vegetation and low ambient light are the norm, melanistic leopards make up a substantial fraction of local populations in some areas, perhaps 40 to 50% in parts of Malaysia. In open African savannas, where a spotted coat matches the dappled light of acacia woodland and the tawny grassland, melanistic individuals are rare because the camouflage advantage reverses. The Turing system built the canvas. Natural selection painted on it. And the genome keeps the record of every brushstroke. Kipling's leopard pressed its fingers into its skin and left spots. The real answer took a mathematician, a half-century of molecular biology, whole genome sequencing of wild felids, and a series of studies on developmental gene expression in cat embryos to arrive at. The mechanism is, in its own way, stranger than the myth. The spots on a leopard are not marks. They are a stable solution to a system of differential equations, written in the language of diffusing proteins, read out during a specific developmental window in the fetal skin, and stored in a genome that has been running the same computation for millions of years. Every rosette on every leopard that has ever lived is the same equations producing the same answer, tuned slightly differently by the particular alleles the animal inherited, the size of the body the pattern had to cover, and the environment in which the animal developed. The black leopard that looks like a completely different animal from its spotted sibling is running the identical patterning program, with one signaling gene turned off. The Amur leopard, with its wide-spaced rosettes, is computing exactly the same equations as a leopard in Zambia, but on a larger body, in a colder climate, with slightly different parameters. The convergence between jaguar and leopard rosettes is not coincidence or shared ancestry. It is two separate genomes discovering the same optimal solution to the same camouflage problem. Alan Turing described all of this in 1952, with no knowledge of the specific genes and no access to a single felid genome. The genome confirmed it. The leopard has been computing its own spots since before the genus Panthera existed, and it has been doing it correctly, every time, in every individual, for millions of years. Subscribe. New discoveries about lost species posted every few days.